Hi, this is Miss Linton, and this is my wonderful Period 1 AP biologist on a Monday morning. Say hi.
Hi.
All right, they're so excited to be here. All right, so we are continuing on in Chapter 9, Cell Cycle and Cell Reproduction, and this is Section 9.4, Cell Cycle and Cancer. So clearly, if we're dealing with cancer, there's a breakdown in the cell cycle and how it's regulated. We agree with that, right? So when we look at cells, okay, normal cell characteristics, like what would a normal cell do? So there's two things they have here: density-dependent inhibition. Density-dependent inhibition. So what does that sound like if you just even didn't read it? So when it gets dense, what are the inhibition would mean what? Stops, okay? So if you look here, and anchorage dependent. So the first thing is, for cells to divide, they have to have a base, okay, that they're attached to. Most cells have to have some sort of base. And if you have a full layer here, then they're not going to reproduce. In this case, they're removing a few cells, and then that gap is filled in. They don't do more than that, okay? They're just filling that spot. And if you look at a cancerous cell, then they're continuing to divide and continuing to divide even though they've covered that layer. So if you look at your notes, normal cell division, A, could Slate tell what A is, and then B, blue will do B, and the last word you need, the two words you need are stop and divide. The two words you need to fill it in are stop and divide. Slate does A, blue does B. Go. All right, so if you have a neoplasm, I gave you that part, that's an abnormal growth of cells, and then that can end in what you're seeing here. You could have some sort of tumor, and there are two classifications of tumors: benign tumor and a malignant tumor. Which one is worse? Exactly. So benign tumor, they can go in, if they find it, you detect it early enough, they can go in and just remove it because it's encapsulated and enclosed. If it's malignant, then it can spread. So on your tumor, you have massive abnormal cells within otherwise normal tissue. Benign tumor, not cancerous, it's encapsulated, has not invaded adjacent tissue. Whereas a malignant tumor, it has the ability to spread. When it yes?
Will the benign tumor always stay like harmless since it's not moved?
That is such a good question. It doesn't mean it always will stay harmless, because it could become malignant. And that's why they talk about early detection being so critical, is to find them early. Like if ever, what sometimes happens if you find a lump, like, and you're like, oh my gosh, I'm scared about it. So what do some people tend to do? They ignore it. And so if you would catch it early, if you would take care of it early, if you have an abnormal mole that's on your body or your skin, then don't ignore it. You know, go early to try to get it taken care of, because you want to do that before it gains the ability to spread. Okay? So on your metastasize or cell separate from a malignant tumor and enter the blood or lymph vessels and travel to other parts of the body. Okay, so let's refer to the cell cycle, and I'm going to let Slate, you handle this one right here. Go ahead. What's going on? All right, so something's going on with the cell cycle. So there's more of them. They're not obeying the rules, okay? There's more of them. And then secondarily, if they then gain the ability to spread to other areas, that's when you're going to have that problem. So when we talk about carcinogenesis, the development of cancer, it's usually gradual. usually is a multi-step process. If you went out today at nutrition and you said, I'm going to smoke a pack of cigarettes, and then tomorrow you got cancer, you know, we've talked about this before, probably people would go, oh my gosh, smoke today, cancer tomorrow. Nobody would smoke, right? But usually what does it take? Years and years and years. There are some cancers, I think cancer of the eye, retinoblastoma, which can happen really rapidly, but most cancers take some time, years, until it's like enough things have to go wrong until it overwhelms that system. Yes?
So
there's a lot of checks and balances, was what I would say. Yes.
This might be later in the notes, but how does like radiation stuff cause cancer?
Well, how does it cause?
Yeah.
Okay, so let's think that out. If you have cancer, right, that means probably you have the wrong cell signals. What are most of our cell signals made out of? Things like what? Could be a hormone. What else could it be? Be more specific of our 4 important organic molecules. What are our cell signals? Usually they're proteins, right? So who codes for the proteins? DNA. So when you have, let's say for instance, one thing, you've grown up in a generation of avoiding the sun, right? You don't want to get sunburned, right? Your parents grew up slathering sunscreen on you, or you had, when you went to the beach, you had a shirt that you wore when you went to the beach so you wouldn't get sunburned. My generation, not so much. We would go outside and put baby oil on our bodies and go in the sun and try to get a tan that way. Okay, we had sunscreen, nobody even talked about it when I was growing up, very rare. And you would get burned. They say now skin cancer is most closely associated with how many times you were burned as a child, sunburned as a child. And you grew up in a very well protected age. Every time you expose that sun or UV radiation, you're exposing your DNA to that. If your DNA gets altered, then the proteins that it codes for could be altered. And that's why you have something like, you know, so it could be a chemical, it could be the sun, something you're exposed to in your environment, something you breathe. It could be a carcinogen in smoking, any kind of chemical like that, or radiation. Changes your DNA, it changes then the proteins. Yes?
But how could something like, so like if you got skin cancer, like a melanoma or something, how could that like get to other parts of your body?
Okay, so at the bottom of your skin, you have what's like your layers. You have different layers, epidermis and dermis. You have what's called a basal lamina. And what happens is normally that would be a barrier. But you can gain the ability to break down the basal lamina. Then once you get into your lymphatic system and all that networking of tubes in your body, or your circulatory system, it's basically like you're jumping on a train and you jump off at another station, and then they're spread again. I think I probably went through this with you at last class, like testicular cancer has a certain pathway, right? It goes testes, and then the next place it'll spread to is your lymph nodes, and then typically to your lungs, and then to your brain. And that's why they say stage 1, stage 2, stage 3, stage 4 cancer is depending on where it's spreading. And they tend to have, certain cancers have certain pathways that they might spread to. And so they gain access to those areas. One process is called angiogenesis, where you develop blood vessels. You call blood vessels that tumor, and it's saying like it needs nutrients, right? Because the cells sometimes will divide, but then they run out of a nutrient supply, so the cell will not undergo cell division. But if you can get a blood vessel to grow to you and dock to your tissues, then it gets more nutrients, and then it can continue to work to spread to other places. Okay? All right, and we are going to go over that too. Okay, let's see where we are in our notes. Cancer is cellular growth disorder that results from the mutations of genes that regulate the cell cycle and loss of control. And then I already gave you this definition right here, carcinogenesis. That was a new word. Okay, so you're going to take turns. Sorry, one of you is going to be cancer, and you're going to ping pong back and forth. So I need Slate, you are cancer, and Blue, you are normal cells. Ping pong back and forth about your characteristic. Role play. I have non-differentiated cells, and then Blue goes. Ping pong. So I spoke to you last class about my mother when she had cancer up at Stanford, and when they would remove cells out of her bone marrow, they would let me be in there to watch how it would go down. And they literally had a microscope they would bring right into the room, all these specialists around. They would go into her bone marrow, they would remove part of her bone marrow, and that was like painful because they make a plug right through your skin. It was a huge needle. She was saying the most embarrassing things, and I'm like, Mom, stop talking, because she had, they give you kind of a sedative. Anyway, it's mortifying. But anyway, they bring out the cells, and then they looked at them right there underneath the microscope. And you could see those cells were so distinct from all the other cells. You could see that those were cancerous cells because they have these hallmark characteristics. When you look at a normal cell versus a cancer cell, you can see that the nuclei are different. You can see that there are maybe more than one nuclei in the cell, and they're abnormally large, and very little cytoplasm. So they're visually distinct. And some other, like the ones that were listed on here, are genetically unstable. Their DNA is in disrepair and it's not getting fixed, and we'll talk about that. They're not controlling the cell cycle. They escape the signals for cell death. They will not go through apoptosis. The DNA maybe that codes for apoptosis, like the P53 gene, is somehow been disabled, and so even though the cell's gone bad, it's not destroying itself. And they can survive and proliferate in other places. This probably, the unspecialization probably helps them with that, because they can move to other tissues and make it there. Okay, let's see. So it all goes back to bad DNA. Bad DNA, mutated genes give you wrong proteins, and that's going to affect the cell cycle and put it out of control. So Slate, take over this slide, just review it, and then Blue, you'll do the next one and explain it. This is a review. Okay, so let's take a look at your notes. So characteristics of cancer cells, they lack differentiation. They are not, what's the word there that we went over a couple times? Starts with an S. Not specialized. Good. They seem what? Immortal. Yeah. Lack differentiation, not specialized. They have abnormal nuclei. They're enlarged or abnormal number or deleted or duplicated portions of the chromosomes. So definitely the nucleus looks different. They do not undergo, what do we call cellular suicide? Apoptosis. Even though normal cell or aged DNA, normally cells enter the cell cycle about 50 times. They form tumors. They're not displaying the density-dependent inhibition. Yeah, inhibition is the word you're looking at. Okay, and then angiogenesis, I've already explained this, so Blue wants to do it though. Go ahead, Blue. Oh, Slate, sorry, Slate wants to do it. Go Slate. All right, so angiogenesis, when tumor cells acquire additional mutations that allow them to direct the growth of new blood vessels into the tumor. New blood vessels into the tumor. All right, so think in your mind, where would you say it all goes back to? The problem's always going to originate with the what? DNA. It's always going to originate with the DNA, because the DNA is coding for the wrong what? Proteins, right? Okay, so then it comes down to two categories of genes here. And I think I kind of introduced this to you when we first started talking about mitosis. Now, that picture up there is general Petraeus, and at the time I made this slide, there were some issues there. So, yes. You recognize this? Yeah. This was played a lot at my house a long time ago. Okay, so proto-oncogenes. Now, what do you hear in that sounds familiar to you? right? So oncologists, right? So proto-oncogenes are genes that are not bad, but because they're in a position of control, when they go bad, they direct a lot of badness. Are you with me on that? Because they're in a position of power. There's so much we could say there. So proto-oncogenes are genes in powerful positions. They're controlling yes to the cell cycle. They're saying, yes, let's go cell cycle, let's go. So if they are sending the wrong signal at the wrong time, then you're going to have too many cells. Well, hopefully you have what are called tumor suppressor genes that are working. That if they do go bad, you have an assassin to take out that general that's giving out that misinformation. So really, cancer is a combination of both of these not working. The proto-oncogene can mutate into what's called an oncogene, okay, and say, go cell cycle, go, and your tumor suppressor genes are like, I got nothing for you, okay, and they're not assassinating those bad cells. So let's start with the proto-oncogene. And why doesn't, I don't know whose turn it is, Blue?
Blue.
Go Blue. Okay, so let's talk about this. Let's say you are, when you turn 16 and you get your driver's license, you're not supposed to drive with a friend, right? Let's say you break the rule and you drive with a friend, okay? Thank God that happened when my boys were in school, because I didn't want to take them to school. Okay, so let's say you're driving with a friend and you're approaching a very busy intersection. You would normally lift up your foot and step on the brakes, okay? But your friend is like, okay? And instead, they step on your foot that's on the gas. And they're like, yeah. And you're like, what are you doing? And you're like, right into the intersection. So you get in a what? Crash, okay? That's kind of like what an oncogene is. It's like stepping on the gas. It's going when you shouldn't go, okay? That's how I want you to think about it. So in this case, here you have your normal cell cycle that you would undergo, okay? And normally you need a growth factor in order to trigger that cell cycle. Yes, we need more cells, we need more cells. But what happens here is even if it's not stimulated by a growth factor, it is still going forward. And on your notes, go to your notes, proto-oncogenes code for proteins that promote the cell cycle and prevent apoptosis, like a gas pedal in your car. So proto-oncogenes, and I don't want you to have this in your head. Proto-oncogenes don't have to become oncogenes, right? You could live your whole life and not get cancer, right? They're your cells that were in positions of power never went bad, okay? A third of you probably will get cancer, okay? Look around the room.
What?
Yeah.
At some point in your life.
Yeah, at some point in your lifetime, yeah.
But like deal with it or like to die?
Well, I'm not saying, well, we'll talk about the percentages there. I have slides on this. Okay? Now, you would want to know, right? You would want to know how many different, if you look at your next ones, how many potential oncogenes do you have? About 100, okay? So there are over 100 potential oncogenes. And they're in different families or groups, and the one that is most, you see it the most is the RAS, and RAS is like stepping on the gas. Good. So normally you need a growth factor, and here's a protein acting as a receptor, which triggers the RAS, which triggers the DNA, that triggers that gene expression where it's going to promote the cell cycle. But in this case, this is a bad RAS. Okay, he has not been stimulated, but he is still sending the signal to DNA, hey, express that gene. Now, this is a RAS protein. Who is he coded for? By the RAS gene, right? So somewhere up here, some other DNA that was bad coded for that bad protein. So something must have happened to that DNA that coded for that. So over 100 potential oncogenes, examples are the RAS gene family associated with lung, colon, pancreatic, leukemia, lymphoma, and thyroid cancers. So in those cancers, they find bad RAS. then you've heard of the BRCA1, right? The gene for what? What is that referring to? Breast cancer, right? So that is where you're predisposed to potentially develop breast cancer, breast and ovarian cancers. That is a type of oncogene, okay, when it's gone bad. Now, our we also have, like the Assassin's Creed, we have the tumor suppressor gene. Slate, could you please talk about that one? Go ahead. So now you're in the car with your friend headed towards a busy intersection. And in this case, what's happening is whenever cells are normally bad, and here your friend is stepping on the gas, right? So you shouldn't be going forward. You shouldn't be going forward in that intersection, but your friend is like, nah, remember, stepping on the gas. And you're like, oh my gosh. So you take your other foot and you're like, oh, and you're going to step on the brakes. Normally, if you would step on the brakes, then that would stop your car. So even though you had a bad RAS in that situation, you were stepping on the gas, the brakes would stop you. But in this case, you go to step on the brakes and you're like pounding on the brakes and the brakes aren't working. So that's why you get in that accident, okay? So tumor suppressor genes normally would take care of that problem for you if you had an abnormal gene promoting the cell cycle. Yes?
So, then how does it mutate?
Well, let's talk about that. What are some things, what are causes? And I actually, here are some, here. You could inherit a bad gene, like if it's a cancer that is heritable, if it's in your DNA, if one of your parents or has a bad copy of that DNA and then you inherit it. That's how people test for the breast cancer gene when it runs in families. They say, oh, my aunt died of cancer, my grandmother died of cancer, and so your mom goes and gets tested to see if she has it. And sometimes what do people do then if they know that they have a high potential for either breast or ovarian cancer? They do what? They go ahead of time and they remove. And they have a mastectomy, even though they don't have the cancer yet. They go ahead and have that, they have a hysterectomy, they have a mastectomy, so that tissue is not even available to become cancerous. Okay? And other causes, if you look here, would be things in your environment. Okay? Smoking, I've talked about, in the sun, certain chemicals, things like being extremely overweight, things like that throws off your hormone cycles. Those can all be, and these right here are all personal choices, right? So what you want to think in your head is some things I can't help. If it's in my DNA, it's in my DNA. I can't help that. That was served up to me. But there are some things I can help, and these would be things you want to try to avoid. So here it would probably be some different things. Pesticides and herbicides, people are talking about that, right? And you get that, I don't use it. I don't know if any of you do. I kind of wonder if I should. Sometimes that fruit wash, you know, that you're supposed to clean off pesticides and things. Does anybody use that in their house? Okay, so that would be something to consider. And you just wash your fruit in it, right? It's like not even a big deal. You're gonna rinse the fruit off anyway, right? Yeah. So do you just like spray it on, or how does it work exactly?
It's kind of just like from a bottle, and you just like pour a little bit, and then you just
rub it on it.
And
Okay.
Wash it off.
Yeah, I would want to compulsively put it in a bottle. I can see myself doing that. Here's the scary one, right? Let's say somebody has an oncogene, and viruses, we haven't learned about their life cycles yet, but viruses are the best genetic engineers in the world. They're all about cutting and splicing DNA. So what if a virus goes in and picks up a proto-oncogene or an oncogene from one person, and then they do what's called transduction and take that gene from one person and give it to another person. You see that? And then that was the method of giving it to another person. So anyway, that gets looked at. What can you do to avoid it? Get your mammograms. Come on, that's kind of funny, right? That is exactly what happens. When you go to get your mammogram, they put it between two plates, and you would not believe how much. I'm just preparing you. But they're good at it, but that is the oddest thing. You're just like, oh my gosh. And it matters not the size. They will smash it, and what they're trying to do is they're trying to get chest wall. And I had a girlfriend, and she did have breast cancer. And so we would go get our mammograms together, and we would do what we called competitive mammogramming. And we'd be in there and be like, no, do it more, I can take it, because our goal was, to encourage one another so that they could check and make sure. And the thing is we don't like to talk about it. It's like, Miss Lynn, that makes me uncomfortable. Don't talk about that. You're talking about breasts, okay? And then what happens is then if you don't talk, these are conversations you need to have. You need to have them at home or with your friends and go, hey, we need to get that checked out. And that needs to be a regular occurrence. If you are sexually active, you need to be regularly going to a gynecologist, right? Because you've heard of the HPV, right? And that is linked to ovarian cancer. You can get vaccinated against that. And so you need to get things regularly checked out to make sure that you are okay. Again, early treatment. If they see precancerous cells on your cervix, they can go and just remove those and get a clean cut on that, and then you don't have to worry about it spreading to other places. Is it uncomfortable when that happens? Absolutely, but death is way more uncomfortable, okay? So that's why you want to go and get those things checked out. And this wasn't even part of the lesson, but I always throw it in here in case people start to ask me, like, how do we do? Okay, so there are tumor marker tests, and so you can go and get tested to see that, like, for instance, prostate cancer. Guys, you don't want to get prostate. Girls, we won't because we don't have a prostate. But it used to be only way they check the prostate is 2 fingers up the anus and in, and they would feel for the prostate gland to see if it was swollen. Now you can just get a blood test and have it done. And I turned 50, and so now they want to check to see if you'll get rectal cancer. And my insurance company sent, I haven't used it yet, but I will. I was going to, but then it turns out I needed to read the instructions. So I thought, oh, I can just do this. But they sent to me in the mail, I want to say a week between when I turned 50, it's like they were like hovering, waiting to ship it, is they have you check your fecal material and you just take a sample of it. Again, I needed to read the instructions. I take a sample of it, and just from that fecal material, they can tell whether or not that you could be potentially from the cells they would look at there if you have rectal cancer. That's way better than me having to take a drink, empty my bowels, which I hear is like horrible. I've never had to do that, but I have heard many times that is a horrible thing. Some of you I know have been checked that way. I haven't had to experience that yet. So then you empty your bowels and then they send a scope up there and look, right? Shoot, harvesting cells just from, that. It seems way better. Again, I have to read the instructions. I was like, okay, I'll go for it on this. And then I pulled it and I'm like, oh, I see what I'm supposed to do. Anyway, so good times. Okay, so they can also check your DNA to see if there are issues. So those are all things you want to maybe do. They can do biopsies of tissues to check to see. And I have not ever had one of these done. I have heard this is incredibly painful. But again, death is more painful. That you can look to see if you have cancer cells. Yeah. All right. So we talk more about the treatments and things. We do come to that later. Okay, so I want to keep going here. We were on the idea of tumor suppressor genes. Okay, so these genes are your brakes. And if your brakes fail, then that's when you end up getting that cancer. So you will have, because we're diploid organisms, we have 2 copies of every gene. If you get just maybe 1 bad copy, you could still have normal development. But if both your copies of that gene are bad, then you've lost that ability. At least you had some brakes here. Here you have no brakes. And if you look at the tumor suppressor gene, if something happens, like you were saying, what could cause it? So some sort of, in this case, they're showing you UV light. So your DNA has somehow been damaged, okay? And as a result, you would normally have an active P53 gene who one of its roles besides cellular suicide is trying to fix it. Remember when we talked about regulation of the cell cycle and you stop and look in G1 as a key point to see, hey, is everything going all right here? If you have something damaged, P53 halts the cell cycle, works to fix it, but then if you can't fix it, then you commit suicide. If your P53 gene is missing or broken, that's when you're going to have problems, okay? So the active P53 gene turns on other genes to stop the cell cycle to wait until it can be repaired. No active P53, you have no brakes. And again, that's right here at that critical G1 stage of the cell cycle. Okay, and this is showing you some of the other facts. If you can't see this, pretty tiny over here to the left, you can see P53 gene. But look at this, CDK. What does that sound like? Cyclin dependent what? Kinases, okay? So somehow you're trying to stop these so that they don't enter into those other stages. So you're going to affect the kinase's ability, and then here you're trying to cause apoptosis. So on your notes, go to tumor suppressor genes, code for proteins that inhibit the cell cycle and promote apoptosis, like brakes on a car. Prevent the cell cycle when the DNA is damaged. There are around 6 identified. RB is involved in retinoblastoma, breast cancer, prostate, bladder, and small cell lung carcinoma. P53 suppresses cancer in 4 ways, in 4 different ways. Do I have a slide that shows that? Donde está? Can I what? Were you trying to say something out loud or just having trouble forming words? Okay. All right, so 4 different ways. 1, it activates a P21 gene that binds with CDKs and gives time for DNA to repair. 2, it activates a group of microRNAs which inhibit the cell cycle. Now, we have not learned too much about this yet because we haven't learned about the process, but do you remember when we talked about DNA replication, right? And we talked about how you use DNA, we talked about transcription, you make RNA from DNA. Welcome. You make RNA from DNA, yes? And microRNAs are small pieces of RNA that bind to the messenger RNA, and if it's bound to the message, it silences the message. You can't use it to translate into a protein. That's what microRNAs might do. Okay, so that's that part. Number 3, turns on genes directly involved in DNA repair and activates suicide genes, caspases, to stimulate apoptosis. And then this is a real tell. This is where the research is getting done. It's estimated that over 1/2 of human cancers due to abnormal or deleted P53 gene. Half of all cancers. All right, so then there's a couple other things. Other origins of cancer in this topic is DNA repair systems fail, which would be really, it's like you're stopping the cell cycle, but they are not getting repaired. DNA repair systems fail. And then telomeres. These are at the ends of chromosomes. And basically, your chromosomes at the very end have some repetitive units that are not, they are not coding for an mRNA that's going to get translated into a protein. It's more about structure. And they get smaller and smaller and smaller and smaller. And that's how your cell knows, oh, stop dividing, because that chromosome has become smaller. Well, there's an enzyme, telomerase, ACE, that's an enzyme, right, who keeps rebuilding the chromosome, and it's not getting smaller, so therefore your cell thinks it needs to keep what? Dividing and reproducing. So telomeres at the ends of chromosomes are not shortening with each replication due to telomerase, an enzyme that keeps the chromosomes at a constant length. Cells with short chromosomes tend to stop dividing, so these do not stop. Okay? All right, that is the end of the cancer talk. There's just a little tiny bit here at the very end about prokaryotic cell division. And on prokaryotes, they have a single circular chromosome located in the nucleoid region, and you already learned about that. And asexual reproduction, offspring are genetically identical to the parent. They do not undergo mitosis. They do something called binary fission. And that single circular chromosome replicates, and then it divides. The cell divides. So binary fission, chromosome replicates, then cell elongates, the cell divides. So if you wanted to compare prokaryotes with eukaryotes, why don't I have Slate You talk about prokaryotes, and Blue, you're going to be the eukaryotes. Go ahead. Talk about the differences. And I completed that chart for you, right? Yeah, okay. Now, are you paying attention at the end of your notes to the end summary and what you must know? You should, yeah. You use them? Yeah. Make sure you do. That, you know what, these would be really good. Remember how we talked about getting into those study teams? Saying we're going to work on the essays and we're going to go through what you must know. You know, that would keep your focus on the most important issues within the notes. So I would recommend that. All right, let's do a few questions, see how you do. And those organized people, you might want to go ahead and open up Chapter 10, Group Shared Notes.
I'm answering the question. You already answered.
Am I? Oh wait, she's yelling at me when I touch stuff I like.
Okay, Rocket Raccoon.
Good job, Prince.
I love Guardians of the Galaxy. Oh, I lost a man.
Why do you have such a short arm?
Oh.
Yeah, could cause cancer. Now, I'm guessing whoever picks C is you're saying would never cause cancer because it has to become a oncogene. I'm just guessing. Maybe that's why they put it. But proto-oncogenes are oncogenes. They've just gone bad. Okay? This is a map. Is it a man? It is a man. Every word on that choice is familiar, right? Yes. So you have to be able to differentiate between what it's saying. Just because there is maybe a true statement, it doesn't mean it's true to the question I asked. Okay, so you got to be careful. So before I hit stop, let's go through it. When cancer occurs, it could be a result of mutations that have occurred. Do you think that's true?
Yes.
I agree. An operational P53 gene? No. Why not? If it's working, you wouldn't have cancer. Apoptosis that has occurred correctly? No, you wouldn't have cancer. Result of the cells no longer continuing the cell cycle? No. They are continuing the cell cycle indefinitely, right? The RAS genes being operational without external signals? Yes. Oh, so few. Okay, discuss right now with your bio buddy. What one did you get right? What one did you miss? Discuss it. Go, Star-Lord. Yeah. Cancer cells tend to be unspecialized. The problem, what they don't do is they don't undergo apoptosis, cellular suicide. Is this a man?
Yes.
Is it a big man?
Yes.
Is it a giant? Giant man.
You just gotta go. You literally wait till she's like,
Yeah. You and I were like, what are we putting that in?
that's
Yeah, all of those. Receptor protein, a signaling protein is off, and a gene, right? You could be sending the wrong message, or the receptor could be doing whatever it wants to do regardless of the message, and definitely a gene's messed up, because it's genes that code for the receptors and genes that code for the message, right? So I'm going to go back to the DNA. Did I just hear, I don't need help on this one?
yeah. Yo, this is gonna be an easy one.
Hashtag things that Jake's.
Jake is always like,
Yay!
Oh my God, you guys.
Ghostbusters #1!
Wonder Woman #4.
I just threw a cell cycle in there for fun. And the end. Okay. Yay! Okay, we need to transition. Chapter 10. Hope you're having great days. Don't stay up too late if you're studying late at night.